Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for UW Extension Cooperative Extension, and on behalf of those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Tom Givnish, and he's from the Department of Botany. He was born in Philadelphia, and then he went to LaSalle College High School, which is a stacked noun, which I love. So that was the high school for LaSalle College. Absolutely. Then he went to Princeton. How far is Princeton from Philadelphia?
50 miles.
Close. And at Princeton University, he studied math undergrad and stayed on there at Princeton to get his PhD in biology. And then he went to teach at Harvard, and in 1985, he came to UW-Madison. Tonight, he's going to talk about one of the cool things ever, plants that eat animals, the carnivorous plants. Is that fair enough? He's going to talk about the Green Empire Strikes Back, the ecology and evolution of carnivorous plants. Please join me in welcoming Tom Givnish back to Wednesday Night at the Lab.
Thanks for those very kind words, Tom. I hope you'll find it a very fun topic we'll be discussing tonight. As you know, most plant species are eaten in whole or part by animals that act as leaf eaters or pollinators or seed dispersers. But carnivorous plants have turned the tables and consume animals as prey. Charles Darwin was the first to recognize and ultimately strongly defend this remarkable fact beginning in 1860. He was walking through an open, acid heath on wet, sandy peat in southern England when he was surprised to see a great number of insects caught by the glistening leaves of the common sundew, which is a low-growing herb. The sticky droplets, secreted by the red glandular hairs or tentacles on each leaf, glitter in the sun like dew, which gives sundews their name. And the sundews entrap insects that range from small flies to larger butterflies and damselflies. Given the large number of sundews present in any given site, Darwin saw that, quote, the number of insects thus annually slaughtered must be prodigious, close quote. Now, Darwin realized that the sticky hairs of many plants, like petunias, kill insects without the plants receiving any benefit other than protection against being eaten by those insects. But he was able to show through a series of careful observations and brilliant experiments, culminating in his classic 1875 book on insectivorous plants, that sundews and a few other carnivores differ in having extraordinary adaptations to entrap and digest insects. His son Francis soon thereafter demonstrated that sundews that caught prey grew faster and set more seeds than those that had not. Given how little nitrate is available to roots in acid wet peat, Darwin argued that carnivorous plants should gain an advantage on those soils by augmenting their nitrogen supply via insect capture. Now, Darwin demonstrated three crucial facts about sundews. First, their tentacles are exquisitely sensitive to pressure and to minute doses of nitrogenous liquids. Secondly, the tentacles, and in some species, the leaves themselves, you can see here, move quickly over seconds to minutes toward entrapped insects, further ensnaring them. And finally, the tentacles secrete fluids that digest nitrogenous substances and then later resorb them. Today, we can visualize these extraordinary movements and secretions and resorptions through time-lapse photography and enzyme assays and microscopy to trace the movement of labeled proteins or amino acids into plant tissues and cell compartments. But Darwin didn't have the luxury of access to such sophisticated approaches, and instead had to rely on his own drawings and a stopwatch, the fate of minute pieces of cooked egg white and meat exposed to secretions, and the microscopic examination of tentacle cells as they resorb the digest. Now, through this, Darwin is able to document or infer the mechanism of prey capture in 7 genera of carnivorous plants. And these included the Sundews of the genus Drosera with 200 species worldwide. I was just actually in the great center of diversity for this genus in Southwest Australia that has about 2/3 of all the species in the world. These Sundews bear active sticky traps with tentacles and often leaves that move over seconds to minutes. And then also the closely related single species of Venus flytrap, Dionea, restricted to a small area in the coastal plain of the Carolinas. These bear snap-trap leaves with 2 lobes bearing nectaries, and as you can see here, trigger hairs nearby. These lobes will close in less than a second when an insect touches at least 2 of the trigger hairs in quick succession. The widely spaced teeth, or cilia, allow small insects to escape while, as you can see, retaining larger prey as the lobes clamp shut. The closed leaf lobes form a tight chamber in which the prey is digested. And the leaf reopens after digestion is completed, resetting the trap. Recent studies have shown that the Venus flytrap actually counts the electrical spikes caused by touching individual trigger hairs with rapid trap closure driven by voltage-regulated ion channels for sodium and potassium and calcium that begin transmitting electrical signals within tens of milliseconds after stimulation. And these cause the leaf lobes to clamp shut, and increasingly so as the prey struggles within the trap, touching the trigger hairs, and it does so through a welter of processes that we still don't fully understand, but almost surely involve water movement and rapid cell wall growth, believe it or not, and a bistable lobe geometry will flip from concave to convex very rapidly. Electromechanical stimuli also release jasminate, which is a hormone involved in responses to insect attack used by most other plants. But in Venus flytrap, it triggers the release of digestive enzymes over tens of minutes to days. Now, while Darwin did not understand the precise mechanism by which Drosera and Dionea rapidly close their traps, he was able to show that some kind of electrical or some kind of impulse traveled through their tissues. Understandably, he became quite enthused with all these discoveries. He considered the Venus flytrap to be the, quote-unquote, the most wonderful plant in the world. And he even wrote to a colleague at the time that, quote, at the present moment, I am more interested in Drosera than the origin of all the species in the world. And so coming in 1860, just after the origin of species, that's saying a lot. By the way, by comparison, Linnaeus, responding to some very early reports of possible carnivory that he received in the 18th century, said it's impossible that if plants were to eat animals, it would contravene the will of God. And he dismissed it out of hand. Now, the waterwheel plant, Aldrovanda, has a patchy distribution worldwide, and essentially is a miniature aquatic version of Dionea, with snap traps that close very rapidly underwater and capture insect larvae and crustaceans, and even small fish. Butterworts in the genus Pinguicula, including about 100 species in the northern hemisphere, have yellow glandular leaves and enrolled leaf margins that serve as active sticky traps to catch small flies. The single species of dewy pine, Drosophilum, from the western Mediterranean, has glandular leaves, but these glands and leaves do not move, and so they form passive sticky traps. By the way, dewy pines are relatives of sundews. They're not pines. Pines are coniferous, not carnivorous. All right. Then corkscrew plants, many of you I'm sure have never heard of these, of the genus Genlesia, 30 species of butterwort relatives from tropical Africa and the Americas, bear strange forked roots. Actually, they're leaves in water or wet sand. And each of them bears, as you can see, spiral grooves with the hairs all aligned up toward where the roots join, and there's a mouth that then leads to a passageway through a series of cones, arranged more or less like a lobster pot or an eel trap. It apparently ensnares protozoa and cyanobacteria, nematodes, microcrustaceans, especially copepods, mites, and unicellular algae. The prey are sort of like shmoos. I don't know how many people remember the old Al Cap cartoons involving Little Abner, but the shmoos were, threw themselves into the frying pan, and these guys swim themselves into the stomach of the plant. Finally, bladderworts in the genus Utricularia, with 250 species worldwide, have unique underwater suction traps. We now know that these bladder-like organs have four-sided or four-armed, glands. So the plant really is four-armed that are inside that pump water out and create a partial vacuum inside, and then a trapdoor shown here, which is perched on a raised threshold. Now, if a small aquatic animal jostles the door or the antennae coming off the door, it slips past the threshold. The pressure outside forces the water in, zips in stage 2, brings the prey in with it, and then immediately snaps shut right after that. Now, as shown by this clip, which shows a crustacean initially swimming behind the ladder, this all happens in a very short period of time. Indeed, only in a few there it was. In just a few milliseconds. It almost looks like a quantum effect. One time it's inside, one time it's outside. And it's a very rapid movement, much faster than the Venus flytrap. And it required the invention of high-speed cameras to study. Digestion begins soon after the bladders snap shut. Now, Darwin's closest friend and colleague, Joseph Hooker, he's the guy on the right, who was the director of Kew Gardens, first described carnivory in pitcher plants in the genus Nepenthes in an address published to support Darwin's ideas. Nepenthes has about 150 species in Old World tropical forests, and its pitcher-like leaves serve as pitfall traps in which insects are attracted to nectaries around the pitcher's rim, or what's called peristome. We'll be revisiting this a number of times later tonight. They slip on that rim or a waxy zone just below it. They drown and then are digested in the pitcher fluid. Darwin only briefly mentioned this plant in his book, as well as New World pitcher plants, Sarracenia and Darlingtonia, that have been studied in very brief form by the American botanist Mark Canby. So these 10 genera, Drosera and Dionea, Eldravandra, Pinguicula, Drosophilum, Genlesia, Utricularia, Nepenthes, Sarracenia, and Darlingtonia, form the carnivorous plant canon. And for many decades, these were the only plants considered to be carnivorous. Remarkably, Darwin and his successors never offered a formal definition of such plants, and that impeded the discovery and study of new carnivores. So in 1984, I proposed a definition based on two criteria. First, carnivorous plants must be able to absorb nutrients from dead animals and thereby benefit in growth, survival, or reproduction. And second, they must have one or more unequivocal adaptations for prey capture, attraction, or digestion. Adaptations for all three of these processes are not required, as evidenced by pitcher plants that lack digestive glands and bladderworts that lack attractants. The requirement for adaptations for prey attraction, capture, digestion is needed to separate carnivores from just every other everyday plant, given that earthworms and nematodes and all manner of microbes are constantly dying in the soil and their nutrients are being absorbed by the roots of plants. It turns out my definition has been widely adopted, and based on it, we now recognize over 800 species of carnivorous plants in 5 orders, 19 genera, and 12 families. Now, carnivorous plants may seem to be oddities, but they raise many fundamental questions. How frequently have they evolved independently? When and under what circumstances? What are the physiological and ecological trade-offs that restrict such plants to some nutrient-poor environments, but not others, like epiphytic perches? And how do the advantages of carnivory differ from those of other unusual mechanisms of capturing nutrients? What fraction of their nutrient budget do carnivorous plants obtain from prey? And how does that affect their ability to photosynthesize? How do traps develop? What shifts in trap structure have evolved within lineages, and how are they related to species ecology? What special kinds of adaptations do plants have to capture and benefit from animal prey? And what kinds of genomic changes have become associated with are associated with the evolution of botanical carnivory? I'm going to address many of these questions today, giving the greatest attention to the costs and benefits of the carnivorous habit and what that applies about where carnivorous plants should be dominant, and some of the really remarkable strategies that carnivorous plants have recently been shown to pursue. Now, over the last several years, several new groups of carnivorous plants have been documented by myself and several other scientists, including species of 2 Bromeliad genera and 1 pipewort, each with pitfall traps, Rurigula from South Africa, which depends on pamirid bugs to eat and digest insects that have been trapped by the plants, and then they excrete their nutrient-rich wastes onto the plant. And the remarkable Philcoxia from Brazil, with tiny leaves just below white sand that trap nematodes. We've also learned that several carnivorous plants have evolved further to capture nutrients by acting as bivouacs or latrines for non-prey animals, or to recruit insects to act as butlers to maintain the traps and keep them functioning. And in some cases, we know now that carnivorous plants have co-opted genes that are normally used to defend against herbivores, like jasminates, or to absorb nutrients in the roots. And these genes are instead expressed in the trap leaves. They're used to transduce signals to move trap components and secrete enzymes, and then resorb digests. So thanks to modern molecular phylogenetics, by which we can deduce evolutionary relationships among species rigorously from divergences in their DNA sequences, we now know that carnivorous plants have evolved at least 10 times independently over the past 2 to 85 million years. Just a few months ago, my colleagues and I used sequence data across all orders of flowering plants to place all the lineages currently known of carnivorous plants in the angiosperm family tree, and we calibrated that tree against time using the ages of 24 fossils. Now, the 3 most recent origins of carnivory over the past 1.9 to 2.7 million years occurred in the monocot order Poales and involve a few species in otherwise non-carnivorous genera of bromeliads and pipeworts. All of them involve pitfall traps formed by multiple leaves rather than the highly specialized pitfall traps formed by individual leaves in more ancient lineages of pitcher plants among the rest of the flowering plants. I discovered one of these new groups of carnivorous plants many years ago in the Bromeliad genus Burkini, Two species, there on the right, of course, have cylindrical rosettes of bright yellow leaves that impound rainwater. Their inner surfaces are coated with a fine waxy dust that readily exfoliates so that insects lose their footing and fall into the tank. The tank fluid is highly acidic, has a nectar-like scent, and contains, as you can see, numerous bodies of dead ants or bees. The leaf bases are studded with umbrella-like absorptive hairs or trichomes that absorb amino acids and mineral nutrients at high rates. The two carnivorous species of Bromeliad dominate several ecosystems atop the rainy and extremely infertile chapuis or sandstone table mountains of southern Venezuela. These are the lost worlds of Conan Doyle, which I and my colleagues visited on foot and by helicopter. We showed that Bromeliad has a greater diversity of specialized mechanisms of nutrient capture than any other genus of flowering plants, including carnivory, nitrogen fixation, the ant-fed habit, potentially a frog-fed tree, and tank epiphytes that capture fallen plant material. We used DNA variation to infer the phylogeny or family tree of Bromeliad and trace its adaptive radiation in nutrient capture strategies. The evolution of tightly overlapping bases that impound rainwater in tanks and absorptive leaf trichomes were the key innovations that led to the evolution of carnivory, shown here in red, the ant-fed habit, shown in blue, the frog-fed habit, shown in green, and epiphytism, shown, as you can see, in blue. Now, 6 of the remaining 7 origins of carnivorous plants occurred in what we call the asterids and caryophyllates among the dicots, or if you know the recent developments in plant systematics, the eudicots. And many of these asterids and caryophyllates bear glandular hairs, apparently defensive in nature. Darwin argued that such hairs were indeed the first step toward the evolution of carnivory. The largest and the oldest of these groups, within the order Nepenthes, includes about 400 carnivorous species, about half the total. Arose 86 million years ago, including plants with a remarkable range of trap mechanisms, including active sticky traps in Drosera, passive sticky traps in Drosophilum and Triphyophyllum, snap traps in Dionea and Aldrovanda, and pitfall traps in Nepenthes, all of them in one order. Of course, they had 86 million years to do this. This phylogeny suggests that carnivory in the lineage arose associated with passive sticky traps, akin to the defensive hairs seen in the non-carnivorous Nepenthes that are sister to the carnivores, and that led to the rise, ultimately, of active sticky traps in the Sundews, snap traps in the Venus flytrap and the Waterwheel Plant, and pitfall traps in Nepenthes. Loss of carnivory in the adult stage of Triphyophyllum in tropical rainforests, and then complete loss of carnivory in several related vines in tropical rainforests. Many species of Nepenthes and tropical vines occur in rainy climates that would rinse away secretions from glandular hairs, rendering sticky traps inviable. And it was also, by the way, provide abundant fluid to fill the pitchers of Nepenthes. The sticky traps of Drosera and Drosophilum generally are found in climates that are substantially drier and hence less likely to wash the secretions away. The second largest lineage of carnivorous plants, including over 300 species in the family Lentibulariaceae, the butterwort family, in the order Lamiales, the mints and their relatives, evolved 43 million years ago and includes highly divergent mechanisms for prey capture. And these include the active sticky traps in the butterworts at the top, the eel traps in the corkscrew plants in the middle, and then the suction traps in the bladderworts. Homology, say the evolutionary similarities among these trap types, remain elusive. The hope is that by identifying the genes expressed in developing and operational trap leaves, we will resolve this long standing puzzle in the near future. Now, pitcher plants evolved in 3 separate lineages. Nepenthes in the order Nepenthales in the Asian, Madagascar, and Pacific tropics, Sarracenia, Darlingtonia, and Helianthera in the Americas, and Cephalotus in southwestern Australia. Their pictures are remarkably convergent in several features, especially in the prominently grooved lip or peristome that arose independently in Cephalotus and Nepenthes. This convergence has occurred despite different parts of the pictures having been derived, being homologous, in other words, to different organs in different lineages. So in Nepenthes and Sarraceniaceae, the picture lid appears to be derived from a leaf apex, a leaf tip, while in Cephalotus, the picture body is derived from the leaf tip, while the lid is derived from the leaf body. So the Cephalotus picture is sort of upside down in terms of its homology relative to the other two families. Let's turn next to the ecology of carnivorous plants, where some of the most remarkable findings in the past several years have occurred. In his pioneering book on insectivorous plants, Darwin devoted only a few lines to their potential ecological advantage, stating that the absorption of animal matter from captured insects explains how Drosera can flourish in extremely poor peaty soils. Considering the nature of the soil where it grows, the supply of nitrogen would be extremely limited or quite deficient unless the plant had the power of obtaining this important element from captured insects. Darwin thus explained why carnivorous plants occur on nutrient-poor sites, but otherwise said very little about the functional or ecological significance of carnivory. It was not until 1984 that my colleagues and I outlined the first cost-benefit model that addressed the context-specific value of carnivory in detail. Over the intervening years, this model has provided a conceptual framework for dozens of studies on the ecophysiology of carnivorous plants, of their ecological distribution, and of the pathways to their evolution. Today I'm going to briefly review and reassess this model, discuss how it can be extended, as we have done just in the last few months, and evaluate the degree to which its assumptions and predictions have been supported. Now, the traps of carnivorous plants provide an alternative source of nutrients, but also involve the costs, the energetic costs, of lures and digestive enzymes, of nutrient resorption, of tract activation and resetting, and of photosynthetically inefficient leads. To determine when carnivorous plants should evolve or be favored competitively, we must compare these costs with the energetic benefits of carnivory. And carnivory should be favored whenever the energetic benefits of a small investment in carnivory exceed the costs. Our 1984 model assumed that advantages in energy capture translate into advantages in competition and thus to advantages in fitness, the rate of production of viable offspring. We also assumed that the initial trap structure was a simple phenotypic step from ancestral forms. No matter what the energetic or fitness advantage of carnivory, it's very unlikely that mutations encoding a complex trap could arise all at once. Given these assumptions, selection within species or competition among species should favor carnivory whenever the initial marginal benefit obtained via carnivory, measured as increased rates of photosynthesis per unit leaf mass, exceed its marginal cost of constructing traps, measured in the same units of carbon. So carnivory might provide, as you can see on this table, 4 kinds of energetic benefits, involving first, an increased rate of photosynthesis per unit leaf mass, second, an increased rate of conversion of photosynthate to new leaf tissue, third, reduction of allocation to unproductive roots, and fourth, partial replacement of autotrophy of photosynthesis with heterotrophy, that is, say, energy captured by eating other organisms. Now, uptake of nitrogen, phosphorus, or other nutrients via carnivory could result in benefits 1, 2, or 3, leading to increased concentrations of rubisco or other photosynthetic enzymes in new leaf tissue, increased stores of nutrients to convert stocks of carbohydrates into new leaves, or decreased demand for nutrients provided by the roots. Many studies show that feeding does enhance the growth of carnivorous plants, especially under nutrient-poor conditions. but few have tested the proposed elevation of photosynthesis by feeding. The strongest support for feeding leading to higher levels of foliar nitrogen and phosphorus, and therefore higher photosynthetic rates in new leaves, supporting hypothesis 1, was provided by Farnsworth and Ellison for 10 Sarracenia species, and by Pavlovich et al. For Drosera capensis. And you can see in both cases that feeding benefits growth, and also in this case, you can see that the benefit is greater in sunnier environments, as you'd expect, given that rubisco is more limiting to growth and to photosynthesis in sun than in shade. Now, we know now, based on a variety of studies, that carnivorous plants get 6 to 100% of their leaf nitrogen and phosphorus supplies from prey. Experimental tests of hypothesis 2 have not yet been conducted. Some support for hypothesis 3 comes from comparative data indicating that carnivores often have smaller roots than non-carnivores. And finally, most of the evidence argues against hypothesis 4. Terrestrial plants mostly obtain nutrients and not carbon from carnivory, although some carbon uptake occurs in a few cases, probably because of the absorption of amino acids. The situation might be different, though, for aquatic bladderworts, which often allocate very little biomass to leaves and can grow in complete darkness if they're supplied with a carbohydrate-rich medium. So let's consider plants of a given size and ask whether a small allocation to carnivory would increase their net growth and thus competitive ability. As the amount of energy invested in carnivory rises to the right, costs should rise proportionally, that C curve. In addition, prey capture, nutrient absorption, and thus the rate of photosynthesis or conversion of photosynthate to new leaves should increase, shown by that top curve, B, rising rapidly at first, but then plateauing with increasing investments in carnivory as the efficiency of prey capture or the conversion of nutrients to enhance photosynthesis or more leaf mass declines. For the moment, we're going to assume that root allocation doesn't vary with allocation to carnivory, and under those assumptions, carnivory should be favored whenever the initial slope of the benefits curve exceeds that of the cost curve. That is, when the benefits of a small initial investment in carnivory exceed the costs. The dashed lines indicate the difference between the costs and the benefits, and the optimal investment in carnivory should occur right at the point that maximizes the difference between the benefit and cost curve, where their slopes, in other words, are equal. Now, photosynthesis is unlikely to rise with allocation to carnivory unless nutrients are in short supply. So the greatest net benefit of carnivory should occur on nutrient-poor sites. Indeed, the usual increase in the growth of carnivorous plants when supplied with prey, seen in 17 of 20 studies reviewed recently, disappeared in almost all cases when the substrate was fertilized. Now, if light or moisture are in limited supply, then they should also reduce the extent by which nutrient capture via carnivory can elevate carbon gain. Let me just go back here. So that's the lower curve in shady or dry circumstances, less of a benefit. In addition, we'd expect that rich soils should elevate photosynthesis, but eliminate almost all further gains that could be due to carnivory. So consequently, we reach the conclusion that carnivory is most likely to be favored on sites that are sunny and moist, as well as nutrient poor, in agreement with the habitats actually occupied by almost all carnivorous plants. And this goes beyond what Darwin had explained in the past. Now, what kinds of habitats are nutrient poor? Wet soils are mostly deficient in available nitrogen, either nitrate or ammonium, due to anoxia, reducing nitrogen fixation, root function, and decomposition. Bogs are precipitation fed and wet, and thus deficient in both phosphorus that usually comes out of the weathering of rock, as well as nitrogen. Our Wisconsin bogs harbor one species of pitcher plant, several species each of the sundews and of the bladderworts. Ancient, highly leached sands are also deficient in phosphorus and nitrogen, accounting for highly infertile soils in boreal and maritime areas that have high rainfall relative to evaporation, supporting a lot of carnivorous plants, as well as lots of carnivorous plants in the Atlantic and Gulf coastal plains of the US on very wet, sandy soils, the tepuis of the Guyana Shield that I mentioned earlier, and the ancient sandstone landscapes of South Africa, shown here, southwestern Australia, where my students and I were just a couple weeks ago, and in Borneo. All of them are hotspots for carnivorous plant diversity. Now, paradoxically, highly calcareous soils, let's just see it. I guess I've skipped over something here. There we go. Highly calcareous soils over limestone or dolomite are cation rich, but often support a lot of butterworts. And we think this may be because they're actually phosphorus deficient, the calcium carbonate complexes with phosphorus. And that may account for the abundance of butterworts on limestone. Based on tissue stoichiometry, the ratios of N, P, and K, most terrestrial carnivorous plants appear to be limited by N or P or N and P. By contrast, aldrovanda and some bladderworts appear to be limited by potassium, by K, which may reflect the large potassium requirements for constructing and pumping water from their aquatic traps. A second prediction of our economic model is that fire over wet, infertile substrates should favor carnivory. Frequent fires can further impoverish soil fertility by volatilizing the leaf nitrogen in the tissue burned. And they increase light levels and soil moisture by removing competing foliage, and doing all that, they favor carnivory. Many carnivorous plants are indeed associated strongly with nutrient-poor, moist habitats that burn frequently. Examples include, as shown here, Drosera and Sarracenia, Pinguicula, and Dionea on fire-swept precosins and low-lying pine savannas of the southern Atlantic coastal plain. Darlingtonia in serpentine fens kept open by fire in the Siskiyou Mountains in Oregon and California. Brocchinia and Helianthera and Drosera on fire-swept wet sandy savannas on tepuis in southern Venezuela. Rorigula and Drosera on fire-swept damp feinboss in South Africa. And Biblis, Drosera, and Cephalotus in fire-swept quangon and reed swamps in southwestern Australia. Seed germination in Biblis, the plant at the bottom, is even driven by smoke. The germination is enhanced by chemicals in smoke. Other forms of disturbance over sterile substrates, including logging, lake drainage, or fractures or landslides that expose bare rock, can also favor carnivores without, of course, volatilizing N. A third prediction of our economic model is that epiphytism works against carnivory and favors myrmecotrophy, that's the being fed by ants. Perches on tree boles and branches are nutrient poor and often sunny, but are also episodically dry and thus unlikely to support carnivorous plants. Only 17 epiphytic species are carnivorous, far fewer than that expected given the number of carnivorous species and epiphytic species overall. And almost all of these epiphytic carnivores are utricularia growing in cloud forests with traps buried below damp moss. Sunny epiphytic perches are more likely to support ant-fed plants than carnivores due to reduced rates of evaporation in the swollen internal chambers of these ant-fed plants via versus the active secretion of nectar and digestive fluids in most carnivores. Indeed, almost all 300 species of ant-fed plants are epiphytes. Next, optimal investment in carnivory, at least in terrestrials, should increase toward the sunniest, moistest, and most nutrient-poor sites. Perhaps the most striking confirmation of this prediction is the decline in the relative size of the photosynthetic keel versus the carnivorous tube of Sarracenia purpurea, the common pitcher plant in New England. As shown here across 26 bogs, keel size increased with the concentration of ammonium in the soil water in these natural bogs, excluding 2 of the sites where nitrogen was not a limiting nutrient. In a bog remote from industrial areas, plants sprayed with different amounts of ammonium nitrate showed a similar pattern, as you can see in the bottom. And pitchers in another bog that received direct doses of both N and P also showed a shift to larger keels with increasing nitrogen. As expected, more keel-rich pitchers had higher photosynthetic rates. Patterns of seasonal heterophyly producing different kinds of leaves also support our predictions. Most carnivores do not produce trap or produce leaves with a low allocation to carnivory during unfavorable seasons when moisture or temperature or light might limit growth, as seen in the pitcher plants, cephalotus, the Venus flytrap, and a number of other groups. Optimal trap mechanism and form should depend on trade-offs associated with environmental conditions, with prey type and trap type. That makes, I think, qualitative sense. Studies on nepenthes over the past decade provide compelling illustrations of this idea. Now, most nepenthes grow in forest openings over highly infertile soils, and they prey on insects. Both climate and prey type appear to shape the pitchers of these species. And it turns out that waxy traps with narrow cylindrical pitchers, narrow apertures, mostly grow in the seasonally dry lowlands, and they capture creeping insects, while traps with wide apertures and large peristomes that are slippery when wet and have sticky or viscous trap fluids. The trap fluid itself is a bit of a trap. Dominate in constantly wet forests, usually at higher elevations, and mainly capture ants or flying prey. Now, the nepenthes is one of 5 genera of pitcher plants that feed to a very large degree only on ants. And this has always struck me as odd. Ants are famously successful in localizing enemies and food sources via scouts, who then return to the nest and recruit workers and bring the whole ant nation down to deal with either. Yet ants, I know from watching all these plants, avidly visit each of these green anteaters. So what gives? One possibility, according to Ulrike Bauer, plant biomechanics expert extraordinaire, is that many species of nepenthes have outwitted those smarty ants by having peristomes or pitcher rims that are slippery only when they are wet. Most pitchers, including nepenthes, have a zone of waxy crystals below the pitcher opening and the nectaries that attract prey. And these crystals readily detach, causing insects to lose their footing, and it turns out the broken off scales contaminate the adhesive structure on the feet of the insects, again causing them to lose their grip and to slide on other surfaces. And as a result, on these plants, the insects slip and fall into the abyss under all circumstances. But Bauer has shown that several nepenthes have highly specialized peristomes that give good traction when wet, but become incredibly slippery to insects when, good traction when dry, they become incredibly slippery when wet. And then it turns out that the ridges and grooves you can see at different scales here keep water on the peristome surface via surface tension, and they prevent the tarsi of the insects from contacting the plant surface, and so the prey just aquaplane into the drink. And during dry weather, the peristomes allow scouts to locate the pitchers and their nectaries and survive, return to the nest, and recruit large numbers of workers to the food source. And If the conditions remain dry, the ants make hay, but as you can see, if things go misty, oopsie daisy, columns of workers slide into the tank. So these traps, which are active only part-time, which at first seems inefficient and unlikely to be favored by natural selection, might actually provide plants with bonanzas based on a crucial weakness in the social behavior of the prey. Truly a bait and switch, worthy of unsavory politicians by these green Machiavellis. Now, it turns out, by the way, by hooking up electrodes to these plants, Bauer was able to show that as the film of water on the peristome increases in thickness, the prey capture rate goes up. So it's high in the morning and the afternoon when the humidity is high, or after rainstorms. She's also found a remarkable strategy, another way of praying in the rain, in Nepenthes rafflesiana, shown here. Its pitchers have lids with waxy crystals on the underside that reduce traction but allow insects to crawl safely under most conditions. But when it begins to rain, some of the ants and flies will seek shelter under the lid, under that little umbrella. And when the raindrops strike the lid, it bends down and then recoils, and the insect is precipitated into the abyss. These movements are much faster than the Venus flytrap, require no energetic input from the plant, and essentially use rain and the prey's own inertia against it. Basically, these are crafty jujitsu warriors. Now, 3 Nepenthes species with giant pitchers at high elevations have evolved into latrines and depend on the feces of tree shrews for nitrogen. And apparently, these tree shrews are all males because they always leave the lids up. These 3 species at low elevation tolerate substantial shade, apparently because they have greatly reduced secretions of nectar and tank fluid and capture nitrogen either by acting as bat roots in this Nepenthes hemsleana, or capturing leaf litter in Nepenthes ampullaria. Nepenthes bicalcarata, though, beats all. It tolerates shade by having very long-lived pitchers. And these are kept functional by ants that they house in little swellings on their tendrils. You see that little twist next to the tank, the pitcher of the plant. And these butler ants basically keep the peristome clean of fungi and algae. And we know from experiments that have been conducted by Clark and others that the presence of the ants triples the amount of nitrogen input to the leaves as a consequence. And it's particularly effective on older leaves. Uniquely, these commensal ants can swim in the pitcher fluid. It sort of makes sense. They're constantly cruising the peristome, which gets slippery when wet. So they're going to slide in. But they evolved the ability to swim and then get back, pull themselves out of the pool. And often they will eat mosquito larvae living inside the pitcher, and in so doing, they prevent some of the food taken up by the plant from leaving the plant and flying off with the adult mosquitoes. These ants, of course, will defecate into the tank fluid. Now, Darwin suggested that snap traps in Dionea, the Venus flytrap, with rapid closure, escape of small prey via spacing of marginal teeth, and digestion in an enclosed chamber may have arisen to capture larger and more rewarding prey that could be immobilized in the sticky traps ancestral to the Sundew family. And here in Madison, Tom Gibson and Don Waller have developed models supporting several aspects of Darwin's ideas and highlighting how important for the growth of Venus flytrap prey capture early in its lifespan is. Mutualistic interactions between ant prey and pitcher plants may further affect and favor the carnivorous habit. Daniel, Joel, and I, a number of years ago, independently proposed that pitcher plants in Cephalotus, Nepenthes, and Sarracenia, all of which prey primarily on ants, may actually be mutualists with their prey. Well, that's easy to say if you're the plant. What's the advantage to the animal? Well, look, plants in nutrient-poor habitats are long on carbohydrates. They're photosynthetic, for God's sake. But they're short on N and P and other mineral nutrients, whereas ants in the same environments may be relatively long on nutrients because of their predation on insects, but short on energy, on carbon. And given the large amount of nectar provided by these pitcher plants that are specialized by ants, and the few individuals from colonies we observe actually being lost to the pitchers, it might pay ant colonies to sacrifice the occasional sister into the well for all the sugar gained, just as it might pay the plants to provide ants with large amounts of sugar if they gain enough nutrients, thereby to elevate photosynthesis by a greater amount. Now, Moon et al. Have recently demonstrated that the North American pitcher plant, Sarracenia minor, earns a net benefit from its interactions with ants. The question remains whether net benefit also accrues to the ants. If ant specialists like Sarracenia minor shown here are mutualists with their prey, they could generate a positive feedback between the potential benefits of carnivory and increasing prey density, with the abundance of each growing larger and larger until they eat Sheboygan. Carnivorous plants should have lower photosynthetic rates and relative growth rates. The advantage of carnivory does not lie in absolutely high photosynthetic rates, but simply in increasing those rates, minus the costs. A meta-analysis by Ellison and Adambeck appeared to confirm these predictions for terrestrial carnivores, but there's a deep problem with these data. The non-carnivores analyzed often grew on rich soils, while the carnivores grew on poor soils. And so this study really should be redone. If you're interested, call me. Astoundingly, no experiments have yet compared growth and carbon uptake by non-carnivores and fed versus unfed carnivores under field conditions, or competitive interactions among the plants. I hope to pursue such an experiment in the very near future, and if you're interested in helping conduct that study, again, please see me after the talk. Just a few last points here. Possession of defensive glandular hairs should facilitate the evolution of carnivory. The preexistence and the co-use of such hairs would reduce the cost curve and consequently reduce the energetic threshold for carnivory. Okay? In accord with one of Darwin's proposed paths to carnivory, 5 of the 10 carnivorous clades appear derived from ancestors with glandular leaves, as exemplified here by the non-carnivores to the left of the red line, plumbago, frankinia, tamarix, their sister to the carnivorous clade of the Nepenthes. Selection for carnivory should increase if sticky traps also continue to protect carnivores from herbivores. And field removal of secretory glands from this Mexican butterwort show that glandless plants sustain 18 times more damage due to insects than the controls. Vengeance shall be mine, saith the insects. Increased ant visitation to Sarracenia minor reduces herbivory and pitcher mortality independent of ant capture. So remnants of defenses against insect herbivory, from which carnivory is thought often to have evolved, may play an important role in maintaining selection for the carnivorous habit. Now, anoxic or toxic soils should favor carnivory and replacement of roots by trap leaves on open, moist sites. Such conditions render root function costly and favor shifting part or all the nutrient uptake to trap leaves above ground. The widespread occurrence of carnivores on sodden soils and their small, shallow root systems support this hypothesis, as does the restriction to heavy metal-rich serpentine soils of Darlingtonia, several species of Nepenthes, and a few species of Pinguicula and Drosera. But assessing the significance of shifts from root to trap allocation using our original model would ignore the complex interplay of allocation to leaves, roots, and traps and their impacts on whole plant growth. And so my colleagues and I have recently developed a new two-dimensional model to explore these trade-offs. It predicts increased allocation to traps versus roots on sites. The more inefficient roots are for nutrient capture, and the greater the response of photosynthesis to nutrient supply. The key role, however, of roots in absorbing water favors a mixed trap and root strategy, even when carnivory is favored. Next, growth co-limitation by multiple nutrients may favor increased root allocation after prey capture. This hypothesis would seem to provide the only plausible explanation for this repeatedly observed paradoxical behavior that as some carnivorous plants catch more prey, they allocate more to roots. But if two or more nutrients co-limit plant growth, then prey capture elevates the level of one nutrient. Increased root growth should be favored as a means of getting the other nutrient below ground. Adamek has argued that prey are such rich sources of nitrogen and phosphorus and such poor sources of potassium and magnesium that prey capture might favor increased root growth to retrieve potassium and magnesium. Last couple of points. Paradoxically, bladderworts in harder, more fertile waters should invest more in carnivory. Well, how can that be? Well, it turns out the harder waters contain more cations and often more phosphorus and nitrogen, but they also contain more CO2, more carbonate ions from which additional CO2 can emerge as photosynthesis proceeds. The greater the concentration of CO2 and bicarbonate, the more photosynthesis should be enhanced by a given amount of prey capture, favoring greater trap allocation in those harder but more fertile waters. Indeed, Susan Nite, working here at the University of Wisconsin long ago, found that trap number in bladderworts in northern Wisconsin lakes increases with water hardness, and the fraction of biomass allocated to traps in utricularia and check lakes increases with the amount of dissolved CO2. And Adamek has shown that in laboratory conditions, addition of CO2 greatly increases allocation to traps, while addition of prey only slightly increases trap allocation. And both of these are consistent with the predictions of our model. Finally, anoxic or infertile soils select against the evolution of tall, woody carnivores. The almost complete restriction of carnivory to relatively short herbs is remarkable. And again, Darwin did not explain that. Wet and fertile soils are favorable for carnivory, but work against tall, woody plants of any kind because of their lack of parenchyma or air channels in the roots and the ease of toppling such trees in wet soils. So I suspect that no matter how widely we explore the earth for new carnivorous plants, we need never fear an Audrey 2 scenario. So to conclude, many of the assumptions and predictions of the cost-benefit model for the ecology and evolution of carnivorous plants in its original and new forms appear to be supported. The most powerful test of our models in the future would entail making them fully quantitative and then comparing quantitative predictions of optimal plant form and physiology with reality. In addition, field experiments on the growth and competitive of carnivorous plants versus non-carnivores in different contexts would be extremely helpful. The revolution in our understanding of carnivorous plants is likely to continue as new genomic data allow us to fully understand the development and operation of traps, assess selection on them in different environments, and quantify the impact that carnivory has on nutrient acquisition, photosynthesis, whole plant growth, and competitive ability. Only then will we fully understand these most wonderful plants which have ensnared our attention ever since Darwin. Thank you very much. I'd be happy to take questions if there are any. Yes?
Going back to the shrew?
Yes, the tree shrew. It's not a shrew. Yeah.
How did the shrew develop that behavior?
What a good question. People have speculated that it may have been that they occasionally visited the pitchers to fish prey out of them. All right, and then of course they probably also may have visited them to go after the sugar secreted by the nectaries on the lid, and that would lead to increased nectar production. The shrews will scent mark their territories. And if a pitcher plant provides a lot of food, it might scent mark it. And the way it scent marks it is urinates or it defecates on it. And you can see how that would select in the plant for bigger lids, more nectars, bigger orifices, and also a perfect match between the snout to vent length of the individual shrews and the size of the pitcher opening and where the lid is. And there is a very good match there. Other questions? Yes?
First of all, thank you. Great talk.
Thank you.
So, all of the carnivorous plants, interestingly, are flowering plants. So
Yes.
what proportion of them are pollinated by insects, and then how might a carnivorous plant mitigate that kind of...
An excellent question. An excellent question. So the question is, how do carnivorous plants avoid eating their pollinators? And about 30 years ago, I proposed that is why almost all carnivorous plants have a leafless scape, a leafless tall stem that separates the trap from the flowers. Now, subsequently, people have found in a few cases that different scents are released by the leaves versus the flowers, and that may also prevent self-destruction. As to why conifers don't do this, that's an excellent question. There are a lot of things that conifers don't do. There, for example, there's only one parasitic conifer. I really don't know what it is, except I would say the obvious, which is to say conifers are all woody. And so I've already explained why woody plants should not become carnivorous. Other questions? Yes?
You mentioned the carnivores rich in nutrients such as potassium, magnesium, and all that.
Mm-hmm.
Would it be of interest to the pharmacological industry?
Okay. So actually they're poor in all those nutrients. They're not rich, but they become richer than the very low levels by eating prey. So I don't think they'll ever be a source of agricultural chemicals. But one thing that just came out with a student who just wrapped up a thesis in my lab was we looked at the coverage by pitcher plants in a Mississippi bog as a function of the amount of, say, nitrogen in the soil. We'd expect that it would just decline, right? The coverage would decline as conditions became more productive. And that generally was true, except at the low end. At the really low end, there was a rise in the abundance of the pitcher plants as soil nitrogen went up. And we think what's going on there, we can't prove this yet, is that when you have pitcher plants invading really poor sites, they catch insects. The leaves ultimately die. Some of the nutrients get into the soil. And of course, now, too much of a good thing is bad, right? And if the site becomes really rich, then taller non-carnivores will probably competitively oust things. Does that answer your question? Yeah. Okay. Other questions? Yes?
I was just wondering what portion of these carnivorous plants are symbiotic with were they both?
Yeah, we don't know for sure because people have not conducted the experiments to show a mutualism in both directions between, say, the ants and the plants. We only have some evidence for one plant species in one direction, not the other direction. So we don't really know. However, there are quite a few cases, non-involving ants, where there clearly is some kind of mutualism involved. So, for example, our pitcher plants lack digestive glands. So how they digest the plant material? Well, partly it's because there are symbiotic fly larvae that live inside the tank. They have evolved the capacity to live inside the tank. There are flesh flies that break up the corpses into smaller bits, and then mosquito larvae that mill it very finely. And of course, there's lots of microbes that are constantly milling those things down. And so there's a benefit to the plant, presumably, in terms of increased rate of delivery of the nutrients. Of course, the flesh flies, the mosquitoes fly away, so they take some away with them, so they benefit too. There's a graduate student at Harvard right now who's looking at nepenthes and quantifying the microbiome He's using next generation sequencing to figure out which groups of microbes are in there, and he finds, depending on where the plants are growing, what they're feeding on, very different microbes, and there's a possibility that they're involved. But again, that has not been demonstrated yet. Other questions? Yes?
Can you talk more about that slide you showed that has the bat inside?
Yeah, so it's a very interesting, more interesting even than just showing that slide. There's one, the Nepenthes hemsliana, that attracts bats that hang out by day. That's their roost. And so they benefit by being out of sight, by being in a humid environment so they don't lose a lot of moisture out of their wings and so on. And they crap in the tent, right? They release nutrients into the trap. Now, what's more remarkable is that particular species has a lid which is cocked back and has a special shape to it, which gives a very distinctive acoustic reply when the bats echolocate. And people have been able to show by clipping off the lid that it's adapted to get the bats to the plants. Yes, Tom.
What are the studies of the roots of these plants show mycorrhizae?
So many of them lack mycorrhizae, many of them lack parenchyma, but some of them do. But I think in general, that's a completely open topic. I've looked and looked through the literature. There's almost nothing written about it. It would be really neat to study, and you could almost instantly make hay. You could really provide.
Agriculturally not important, though.
Yeah, more's the pity, I suppose. Yes.
Do we know if carnivorous plants form mycorrhizal relationships?
Well, I was just saying that many of them lack mycorrhizal fungi, but it simply has not been surveyed. So I wouldn't want to make. I did not make a pronouncement because I don't think we know enough. Okay. So, for example, look, one of the groups occurs in, actually two groups grow in the Ericales, which has very strongly developed mycorrhizae, but they haven't been looked at yet. Yes?
If the plants are given synthetic nutrients instead of natural nutrients,
yes,
are they able to digest that?
Well, Darwin showed that long ago, that he, for example, provided them with little agar blocks, and they could digest that. Now, it's also the case that if you add just ammonium nitrate to the tanks, as in the experiment I showed you, they can benefit from that. There is a case, however, that many of these carnivorous plants cannot stand very high levels of nutrients in the soil. So in other words, if you give them a little extra nutrients in the soil, they will grow faster. Of course, other plants will grow even faster, we think. But if you add too much, they die. And this is seen in a number of plants that are adapted to extremely infertile soils. Presumably, their membranes and their roots are adjusted to really soak up the nutrients at high rates. And if you provide too much, it's like, giving a Polynesian constant source of carbohydrates. They're going to go diabetic and die. So I don't know that that's the case. That's suggestive, though. Does that answer?
Yes.
Okay, very good. A couple more questions. Yes?
Yeah, I'm just a little bit confused. What types of poisons do these carnivorous plants use to kill other than aphids?
Right. Okay, so I didn't really mention anything about that. Many carnivorous plants, but not all, have glands that secrete digestive enzymes. And basically they immobilize the prey and then they work their way on it. Now, they kill, like the Venus flytrap, will kill the prey not by directly digesting it while it's alive, but the secretion of all of the glands inside the closed leaf drowns the damn thing, right? And in pitcher plants, we know that our pitcher plants, the Saracenia purpurea, there's a wetting agent in the tank fluid that increases the rate at which water penetrates the spiracles, the breathing apparatus of the insects. And in addition, the plants secrete an alkaloid called coneine that is a narcotic and sort of puts the prey to sleep as it drowns. Last question, yes?
Can you talk a little bit about the fossil record for carnivorous plants, if there's any?
There is one and only one fossil, and it was published a couple of years ago by a German group working in Scandinavia, looking at, they found it in amber. They found it in amber, and it's clearly Ruridula, because Ruridula has like 3 different kinds of hairs on its leaves. It's unmistakable. And the weird thing is, today, Ruridula is only found in a tiny portion of Southwest Africa, which was never associated with Scandinavia. So clearly it had a wider range upon a time. Yeah, it could be, yeah, exactly. Slashing and burning all the Ruridulas. Yeah, exactly. Well, thank you very much. If you have any other questions, I'd be happy to entertain them afterwards.